Last slide, are you able to hear me?
Yes.
Okay. we have, I've said we have three layers of the cerebellum. One is the molecular layer, comprising of glial cells, comprising of basket cells, and the basket cell synapse with the Purkinje. We also have the parallel fibers are axons of granule cells. The granule cells originate from the granule part, and then when they reach the molecular layer, they bifurcate into two, so that it forms something like a T. Each of those T extends for a long part, some millimeters, and therefore synapse to the Purkinje. Within the molecular layer, we also have the dendrites of the Purkinje, forming that arborizations of the Purkinje. Then we have the horizontal cells, stellate cells. We have stellate cells which are running horizontally. That's what we have in the molecular layer. So this is an association area. There's a lot of synapsing in this area. The next layer consists of a single layer of cells that we refer to as the Purkinje cell layer, or also ganglionic layer. This is the cell body of the Purkinje. It is flask-shaped cell with a single axon, thin axon, projecting into the deeper parts of the cerebral cortex. Cerebellum, sorry. It's the only output from the cerebellum. The next layer here is the granule layer. And the granule layer consists of granule cells. The granule cells, so the nuclei of the granule cells are located here, the large granule cells, which are stellate shaped cells. And there are dendrites, synapse in these places here, they seem to be empty. The synapse in these places, together with the mossy fibers, So these areas are what we call cerebellar glomerulus. So the dendrites of the granule cell synapse in these positions here, in the molecular layer, the axons project to the molecular layer. When they reach the molecular layer, they bifurcate. And run parallel to the surface as parallel fibers, synapsing the several Purkinje cells. Within the granule layer, we have the axons of mossy fibers. So the mossy fibers will synapse at the cerebellar glomerulus. We also have some Golgi cells, and these ones here, some of the nuclei are in the cortical part. The nuclei are in the cell bodies are in the cortical part. These axons extend to the granule cells, and they synapse at the cerebellar glomerulus. Synapse with mossy fibers and granule cells. Those synapses, both from the basket cell, are inhibited. But the synapse, the granule cell which extends to the molecular layer is excitatory. So it synapses with the dendrites of the Purkinje, and also will synapse with the dendrites of the Golgi cells. So this is what we have as the granular layer of the cerebellum. But like I said, the only output will be from the pyramidal cell. Apart from this, arising from the pons and also from the cerebellum, from the cerebral cortex. We have fibers which are referred to as climbing fibers. The climbing fibers synapse with the Purkinje. Each Purkinje will synapse with one climbing fiber. The cell bodies of the MOSI fibers are actually in the pons as the pontine nuclei. They are the pontine nuclei. Axons are the ones which extend to the cerebellum, now synapsing with both the Golgi cells and the granule cells at the cerebellar glomerulus. Within that cerebellar glomerulus, we have some specialized glial cells that form something like a capsule around the glomerulus. This we refer to as the vallet type of cells. Vallet cells. This is just an example to show the cell bodies. This is the ganglionic layer. And then at the molecular layer, we have very few cells. In the granule layer, we have a lot of cells, with a lot of these spots being areas for cerebellar glomerulus. This is the cerebellum. You will notice here that we have an outer part which is having very few cells. That is the molecular layer. We have the granule layer, and in between molecular layer, we have a single layer of cells that we refer to as the ganglionic layer, comprising of flask-shaped cell bodies of pyramidal cells. Is exactly what you have again, of showing the various parts. This is just to illustrate a single axon from the cerebellum. You'll be able to see this slide in our practicals. This is the cell body of the Purkinje forming the ganglionic layer, and these are the arborizations of the Purkinje, arborizations of the dendrites of Purkinje, forming something like a dendritic tree. If you remember when we were talking about the area gelatinosa in the case of the spinal cord, we also refer to as like something like dendritic trees. So it's similar to what we have here. And then this is the granular layer, comprising of granule cells and the white matter area, the medulla. This is to illustrate the arborizations, very large dendrites of the Purkinje, to illustrate the parallel fibers. These ones here are running parallel to the surface, and also to illustrate the basket, dendrites of basket cell. You can see this black, the basket cell is in the molecular layer, but its axon forms dendrites around the Purkinje, inhibitory dendrites around the Purkinje. That's why it's called basket cell, because the dendrites form something like a basket, almost something like a nest of a bird, in which the cell body of the Purkinje can be representing the egg. That's why it's called basket cell. Again, a similar structure, basket cells, you can see very clearly here, dendrites of the basket cells, and then the granule cells and dendrites of Purkinje. Fine. We've seen the three layers of the cerebellum. We have both inhibitory and excitatory. So what is the role of the cerebellar glomerulus? Cerebellar glomerulus is a modulating type of synaptic conduct. Information coming from the basal ganglia, information coming from the part of the basal ganglia, and then part of the pontine nucleus, and also from the cerebral cortex, being sent in by climbing fibers and MOSI fibers specifically. So at the MOSI fibers, we have then the granule cells synapsing And then we have the Golgi cell synapsing, and those ones are inhibitory. So the output, the excitatory part from the granule cell that stimulates the Purkinje, and also the synapsing between Purkinje and the glanding fibers, ultimately the output depends on the summative effect of all those. Output from the cerebellum depends on summative effect of all the afferents, specifically the Purkinje cell, which is supposed to be the only efferent from the cerebellum. So what happens when you have cerebellar dysfunction? What are some of the dysfunctions of the cerebellum? One, we might have demyelinating diseases. Where myelin sheath sort of breaks down, and you know the function of myelin sheath is for fast conduction of the impulse. So if it is demyelination, then that is slowing down. That fast conduction is coded type of conduction, coded type of information. So if it is slowed down, again the brain will interpret that as a different thing altogether. So that is important. And also, what are some of the things that can cause problems? Alcoholic cerebellar degeneration. You have seen alcoholics normally having problems with holding even a cup. So we have demyelination, we have cancer, neoplasms can originate from cerebellum or elsewhere in the brain, but affect the cerebellum. We can have the cerebellar vessels, the supplied blood vessels supplying the cerebellum being occluded, aneurysms or embolisms, and those will lead to hypoxia and damage to the cerebellar cells. So all this can cause some changes in the motor activity. Some of those changes will be something like ataxia, intentional tremors. You want to hold something and you start shaking. Okay, that's intentional tremors. And you have seen alcoholics usually, they can hardly hold their glass, they shake very much. Dysmetria is where you are not able to judge the distance. You judge the distance, but as you try to move towards that object, you find that you are not judging it accurately. Because again, the slowing down of the transmission of the impulse is causing that. That's dysmetria. Now, in dysmetria, for example, you might want to touch your nose, but as you move your hand to touch the nose, you find you are touching your face because this coordination is not there. Decomposition of movement. You break down movement. You have seen people who are drunk. I'm using drunkards because it's a clear example of what you normally notice. You see them the way they walk. They walk not as a coordinated movement, but they walk in and breakdown sort of movement. Scanning speech. This is motor. Scanning speech. You find somebody with a slur, pulling words and so on, and yet he was okay before. So that again could be also from the cerebellum, because our motor, our skeletal muscle, the occipital muscle, originate from the occipital myotomes that supply the tongue. That is the hypoglossal nerve supplying the tongue, and therefore those tongue muscles are not working properly because of the innervation, and because the innervation from the hypoglossal, the information is coming from the cerebellum to fine-tune that movement, and also from our speech areas in the brain. Our Broca area, the Wernicke areas, all those are giving feedback. Like I said, cerebellum receives information from various parts of the brain. Nystagmus, that's movement of the eyes. You'll be able to look at that when you're looking at the visual system. Then we have problems with equilibration. equilibration, staggering. Equilibration is maintenance of tone. Staggering, you're not able to coordinate your movements and therefore you stagger. Or muscle tone is completely loss of muscle tone. So when your muscle tone is lost, again, because of that feedback, it is going to the brain, but the cerebellum cannot give feedback to the muscles to contract. The feedback is there from the musculoskeletal apparatus, Golgi tendon apparatus, vestibular apparatus, but the cerebellum cannot give that command to the muscles to contract. Or, as the command is going, it is slowed down because of demyelination. So we need to note that. Well, this is just a repeat of what we've done, but showing clear picture of what we see. We have the molecular layer, we have the ganglionic layer, and the granule layer. That is what we will cover today. The next class we are going to have will be a brief mention of the diencephalon, and then the derivatives of the diencephalon, and then we'll move to the special sensory systems. At this stage, I'd like to invite any questions. If you have any questions, please put them in the chat. Any areas which are not clear, put them in the chat. And apologies for the interruption, network problems. For reference, first I'd like you to read the histology guide. That is one way you can look at, and any neuroanatomy book. Histology guide and any neuroanatomy. For nursing, if you can lay hold of Marriott, you can look at Marriott. The second question is the climbing fibers. They are not cells. The question was, did you say the basket cells, climbing cells are afferent? The climbing fibers, they are not cells but fibers, are afferent to the cerebellum. They take the information to the cerebellum. Basket cells are within the cerebellum. So they will act like interneurons. The cell bodies are in the cerebellar molecular layer of the cerebellum. Axons then synapse with the Purkinje. Axons synapse with the Purkinje. The dendrites will be synapsing with the climbing fibers. Will be synapsing with the granule cells, the parallel fibers of the granule cells. They will also be synapsing with the Golgi cells. The Purkinje, let me just go back and show that particular picture. You had seen before. If you notice here, this is the cell body of the Purkinje. Around this cell body of the Purkinje, you have these dark structures. These dark structures are axons, are dendrites, terminal dendrites of the basket cell. Terminal dendrites of the basket cell. So a single axon of the basket cell then breaks out to form several dendrites that come to the base of the Purkinje, cell body of the Purkinje, like a basket. Just almost around that base of the Purkinje and synapse with it. Most of this synapsing is inhibited. So the last question I think I've been able to answer. Any other question? Okay. The last question as to the synapsing. I think physiologists will be able to tell you with synapses. Biochemists will tell you the type of neurotransmitters. We have the GABA, GABAergic neurotransmitters. The mossy fibers, synapsing with the mossy fibers synapse with granule cells. So the mossy fiber synapsing with granule cell is excitatory. The granule cell then synapse forms the parallel fibers, which synapse to the Purkinje, the dendrites of the Purkinje. That is excitatory. The basket cell synapses with Purkinje. That's inhibitory. Now, the last question is one which several books have been written about: the conscious and the subconscious brain. Well, you talked about unconscious, not subconscious, unconscious brain, where you are not aware. Remember that visceral activity, your brain beating of the heart and so on, all that is regulated by, you don't decide. It's not voluntary, it's involuntary. But when it comes to the mind, because the question is specifically on the mind, there is no specific part of the brain that is responsible for the mind. Okay. There's no specific part of the brain that's responsible for the mind. However, if you want to put like the frontal lobe, prefrontal cortex, which you make decisions and so on, as part of your conscious, because you decide, and the further parts are subconscious. However, a lot of the parts of the brain you cannot classify mainly as conscious or subconscious. The output from the cerebellum is to fine-tune movement. Most of the output from the cerebellum is mainly inhibitory. The cerebral cortex remember, sending information to the muscles, so the muscles can contract. But cerebellum modulates that information. So what happens if you cut the cerebellum out from it, corticospinal tract, without any modulation? Then the muscles will contract. They'll contract. You'll have very jerky movements. Muscles will contract. But in terms of fine-tuning the movement, that is the responsibility of the cerebellum. Last question is, what about the practical? We are going to have a practical on the midbrain, cerebellum, cerebral cortex, limbic system, and reticular formation. So the class reps should organize when you can have that practical and let me know in advance so that we can alert the technicians and also see whether the lab is available. So the class reps, please get in touch with me when you feel you are free, and also get in touch in advance so that, because currently there are paintings in the department, so we need to know when we can be able to use the lab. And also availability of the technical staff. Thank you. I've said the next class will be on the sensory systems, that is the visual system, and also the auditory system, the gustatory system, and the olfactory system. So read about them so that we will discuss. If you have any questions, also during the practical, you can be able to ask them, or just before the next session, you can be able to ask them. Like I said, we are going to revisit some of these parts of the brain as we look at the visual pathways, auditory pathways, and gustatory pathways, so that you can be able to integrate the information and realize that most of the central nervous system is for integration of the information, and for the brain, it's mainly higher brain functions. Thank you. We'll meet when the class rep decides, but I suggest we do the practical first, and then we can go to the next session.